We tend to think of our genetic code as this really strict, unchangeable rulebook. But what if I told you that the most amazing stories in biology, and even in your own kitchen, don't come from following the rules, but from breaking them? That's what we're diving into. I mean, really think about it. Have you ever wondered why the bananas you get at the grocery store are just perfectly seedless? Or how on earth we managed to create a type of wheat that's so strong, so robust, it can feed billions of people? These aren't just happy accidents. They're the result of some massive, rule-breaking genetic events. And we're about to figure out exactly how. So let's start with this first big idea, the superpower of having extra chromosomes. The answer to our questions begins with a concept that sounds like it's straight out of a science fiction movie: organisms getting entire extra sets of their genetic instructions. The technical name for this superpower is polyploidy. Now, most animals, including you and me, were diploid. That means we have two complete sets of chromosomes, one from mom, one from dad. A polyploid organism, though, it has more than two. Maybe it has 3 or 4 or even 6 full sets. It's kind of like having multiple copies of your entire genetic library. And you might be thinking, okay, that sounds like a super rare genetic glitch, right? Well, not at all. It turns out that a whopping 30 to 35% of all flowering plants are polyploids. This isn't a bug in the system for plants. It's a major evolutionary feature happening all around us. So how does this even happen? Well, there are two main ways. On one hand, you have autopolyploids. That's when the extra sets of chromosomes come from within a single species. This often leads to bigger plants, bigger fruits, but they can have some problems with fertility. And then you've got allopolyploids. This is where it gets really interesting. This is when two different species manage to crossbreed, combining their unique genetic libraries. This is how you can create entirely new species. The story of our bread wheat is the perfect example of this. It's a crazy family history. It all started when two different wild grasses crossed and they made a hybrid that was sterile. But then, something amazing happened. A spontaneous doubling of all the chromosomes. Suddenly, every chromosome had a partner, and boom, the plant was fertile. But it didn't stop there. That new plant then crossed with another wild grass. And guess what? The same thing happened again. Another chromosome doubling event. The result is the bread wheat we have today, an allohexaploid with 6 sets of chromosomes from 3 different ancestors. That's why it's so incredibly hardy. And what about those seedless bananas? Yep, you guessed it, polyploidy again. The bananas we buy are triploid, meaning they have 3 sets of chromosomes. That odd number is a complete train wreck for meiosis, which is the process that makes reproductive cells. The chromosomes just can't pair up correctly, the system breaks down, and so no viable seeds can form. It's a genetic mistake that we've cleverly turned into a convenient, delicious snack. Okay, so for plants, having extra sets of chromosomes can be a legitimate superpower. But what happens when the mistake is smaller? Like, what if you just gain or lose a single chromosome? Well, for animals, this is where the story shifts from a superpower to a very dangerous numbers game. This condition has a different name, aneuploidy. So instead of having whole extra sets, an individual just has one extra chromosome, or one missing one. It's usually caused by a little error during cell division, called non-disjunction. It's basically when the chromosomes fail to separate like they're supposed to, leading to an egg or sperm cell with the wrong number. And this isn't some abstract concept. In humans, aneuploidy is the genetic basis for several conditions you've probably heard of. For instance, having an extra copy of chromosome 21 results in Down syndrome. Other examples involve our sex chromosomes, leading to conditions like Turner syndrome or Klinefelter syndrome. It just goes to show how incredibly sensitive animal biology is to the right dosage of genes. And this really highlights the huge difference between the plant and animal kingdoms. That full-on polyploidy, which gives plants their superpowers, is almost always lethal in animals. Even the interspecies hybrids that actually survive, like a mule, which is a cross between a horse and a donkey, are sterile. The chromosomes from 2 different species just can't get on the same page to make the process of meiosis work. All right, section 3, genes that stick together. Up until now, we've been talking all about the number of chromosomes. But what about the genes that actually live on those chromosomes? Turns out they've got their own way of breaking the rules.
You might remember Mendel's law of independent assortment from biology class. It basically says that genes for different traits like eye color and hair color are passed down independently. And that's true when the genes are on different chromosomes. But what happens when they're on the same chromosome? Well, they're physically connected. They're linked. They tend to get inherited as a package deal, like passengers on the same bus. And this completely messes with those classic inheritance patterns we learned about.
But here's the twist. That linkage isn't foolproof. During meiosis, chromosomes can actually tangle up and swap pieces of themselves in a process called crossing over. And a brilliant young student named Alfred Sturtevant had this incredible light bulb moment. He thought, wait a minute, what if the chance of a crossover happening between two genes has to do with how far apart they are? He realized that the frequency of this recombination could be used to literally measure the distance between genes on a chromosome. Section 4, mapping our genetic code. And that one revolutionary idea, that recombination frequency tells you the distance, was the birth of genetic mapping. To see how this works, let's look at a rabbit cross that involves two linked genes, one for coat spots and one for hair length. So out of 158 baby rabbits, the vast majority looked just like their parents. You had 75 spotted rabbits with short hair and 66 solid-colored rabbits with long hair. But there were these two small, interesting groups. These were the recombinants, the ones where a crossover happened. There were 10 spotted rabbits that had long hair and 7 solid rabbits that had short hair. Okay, now for the surprisingly simple math. We just take the number of recombinant offspring, those two small groups, the 10 and the 7, which gives us 17. Then we divide that by the total number of offspring, 158. What do you get? About 10.8%. And here's the beautiful, elegant part. Scientists basically just decided, let's call a 1% recombination frequency 1 map unit. So that 10.8% we just calculated means that the gene for spots and the gene for hair length are 10.8 map units apart on that rabbit chromosome. By doing this over and over for thousands of genes, we can literally build a detailed map of an entire genome, all based on how often genes get separated. It's genius. So, from seedless bananas to the very map of our own DNA, it's pretty clear that our genetic code is not a static blueprint. It's a living, breathing, dynamic story. And understanding the exceptions, the times the rules get broken, is just as critical as knowing the rules themselves. Which really leaves us with one last big question. What new rule-breaking events are happening as we speak, writing the next chapter of life's incredible genetic story?